works to do that. So, let's see, it's 12:02 my time, which must mean it's 10:02 Second Life time. So, seems to me like a good time to talk. Apologies, I had originally envisioned a YouTube live stream today from my lab doing demonstrations, but I couldn't get that to come together. Um, it's been busy. I'm spending a year being chair of the math department at my school. So this week was quite busy. We actually have a week off around Thanksgiving next week, so there was a lot of last minute stuff to do. And evidently there are issues with streaming into Second Life at the moment with YouTube, so probably best. So what I decided to do was to do a reprise of a talk that I had given in 2016, and that's like 9 years ago, and I can't believe that it's been so long, right? And it's basically something that I've always found very interesting. I'm going to actually talk about some silicon chemistry. I'm going to talk about more high temperature and high oxidation state silicon chemistry. So um, things that might be more susceptible to living under an oxygen atmosphere or at high temperature. Think the Horta. I also want to pitch this as like advice to science fiction authors, right? So um, I'm gonna take it away now. So basically, let's see. This is the old viewer, so I have to use that. So today's itinerary, we'll talk a little bit about some sci-fi tropes, talk a little bit about what life is anyway, and like what does our type of life do with silicon anyway. What's the big deal about silicon? If you look at the periodic table, it's sitting right under carbon, should have the same chemistry. Why can't we just do a search and replace? Well, we can't, and I'm going to tell you why. I'm going to talk about high temperature-based silicon life. And of course, I will have gratuitous cat pictures. This is the original talk, so these cats are much older now. Seth is about 12 years old at this point, and he gets very sleepy. Some of these cats actually have passed away, so sorry about that. But yeah, there's always new cats. Alrighty, yeah. So I think a lot of people first get their introduction to silicon-based life from Star Trek, specifically an episode, "The Devil in the Dark," the Horta, and in fact, there, you know, 12 years ago, there was an action figure of Mr. Spock doing a mind meld with the Horta, which actually kind of looks like a meatball um, off of a pizza with some kind of low-grade mozzarella cheese stuck on it. But all of those ingredients would be carbon-based. We also have a lot more in Star Trek. When you think of nanites from the Next Generation, all of those little critters would have been silicon machines. Again, from the Next Generation, they had a lot of silicon life in Generation. There are silicon chip life forms that, basically were taking over the ship. Who doesn't take over the ship? And their most memorable phrase to me was their description of humans as ugly bags of mostly water. I feel that way more and more these days. Um, so, yeah, Dune, of course, is, interesting. No one comes out and says it, but the sandworms are suspiciously sensitive to moisture. And that fits right into some forms of silicon type chemistry. And basically they have developed an ecosystem where the sandworms are happy to roam around in the desert because their larval stage actually seeks out an encapsulate water. Um, and another type is hey, Silicon Life would be an AI, right? And 9 years ago, that was much more distant as a science fiction concept, like Neuromancer and the whole genre. But these days we have lots of active discussions about artificial intelligence, right? And then you go way back to the 1940s, the frigid-blooded poison breeders from G.E. Doc Smith's Lensman series and things like that. So it was huge, right? And Tag, I'm going to be telling you about this particular stuff. So the silicon is not a cut and paste replacement. It just won't work. But this is about advice for science fiction writers who would benefit from something that might be a bit more plausible. So let's go on. Next one. And back to birth. And so there's lots of um, literature, actually in the scientific literature, on this topic. There's a couple of arguments in the literature that silicon-based life could happen, and then there is no silicon-based life in the solar system. Chemistry reviews talked about it. There's been talk about using a zeolite. I'll show you some zeolites later on as enzyme mimics. But here's the thing: there's a lot of references that in 251 in 2016. I did not get a chance to update, but there's a lot of references and a lot of thought about this particular type of topic. Right? So looking at this thing. Basically, silicon-based life in the solar system, kind of thinking about this particular paper is going to be highlighted more in part two. Silicon-based life on Titan, for example, or, rigid, cold life where silicon-hydrogen bonds and silicon-silicon bonds would be more stable. But I'm going to talk about something a little bit different there. There is life, and where can I get one? So NASA added a definition of life, a self-sustaining chemical system capable of Darwinian evolution. That's kind of a definition I'm going to go with. And when you keep going back with that, there's been some research that, or even just ideas about origins of life on Earth, involving templates involved in clay. That, of course, is a silicate. Clay is, basically silicon, oxygen atoms with various charges and various other, usually metallic elements, forming binding the various units together. Right? That's what the whole idea is about 50, 60 years old now. And there's been some references that I can share with you later on about these sorts of things. Yeah, so here's an example of silicates. I think I've got my handy dandy little pointer up. And essentially, let's actually talk about what silicates might be. I edit this and I move this box in front of us, right? So in front of the screen. the chemistry, tetrahedra are wonderful things. And silicon usually is found at the center of a tetrahedron of oxygen atoms. And a really convenient way um, of fashioning a tetrahedron is to take a cube. As you can see, the cube that I've made kind of transparent. And you join the opposite corners on the top face and the alternate set of opposite corners on the bottom face. And then you join all of those essentially by lines. Each corner of the tetrahedron will be where an oxygen atom is, and lurking in the center, right? So be their actual center of the tetrahedron and the center of the cube in this case, would be a silicon atom. So you could represent more complicated structure, move this one out of the way, as series of tetrahedra that are joined together, right? So this one, for example, this ring is a let's see, it's silicon 6O18, and it's got a charge of overall 12 minus. So there's probably something like lithium ions or sodium ions around it. This is, a particular way of putting together a silicate. Why is it a silicate instead of just silicon dioxide? It's because we have more oxygen attached to the silicons, and the oxygen becomes essentially attached to the silicons as oxygen ions, let's say, right? O minus or O2 minus, so that it brings charges with it. So yeah, these things build up into the structures that we see here. This is a, let's see, Y is an abbreviation for a particular type of clay. They've embedded some platinum in there, and the platinum can actually do some action to organic molecules. I think they're basically taking ethanol and probably turning it, or they're oxidizing it. Yeah, they're probably just oxidizing it. So silicates can certainly promote interesting chemistry happening. So, and anyway, why silicon? Why would anyone be interested in replacing carbon by silicon? Well, looking at the plot in the periodic table, carbon is in the second row of the periodic table. Silicon is directly beneath. And what that means is that from the outside, a silicon atom looks very much like a carbon atom in terms of, how the areas where electron density can go are arranged. So, if I were going to say that in a more chemically way, I would say that the silicon orbitals bear a striking similarity to the appearance of the carbon orbital. Okay, and then there's germanium, but yeah, I think that's a reach for germanium-based life would be a real reach. So, speaking of silicon in terrestrial life, there's a lot more silicon on this planet in the crust than there is carbon. So, silicon forms 28% of the crust. Carbon is 0.03. That actually tells you something about the versatility of carbon as a matrix for life as we know it. It's not just the availability of a particular type of atom that is key here. It is what you can do with it. And carbon you can certainly do a lot more with. There's actually more iron, manganese, and even vanadium has about on the same order of magnitude abundance of carbon. Right? Our quote unquote biochemistry can handle silicates. I mean, when I say our, I'm basically calling us like cousins to diatoms, but that would be a closer relationship to us having a silicon life. Okay? So there are plenty of plants that you have to manage silicic acid. So that is a molecule, or that is a compound that contains silicon. It's basically um, silicon dioxide that has had some OH groups attached to it. Um, Diatoms basically build their shells and skeletons out of silicate, basically they have glass skeletons. So there's no reason why terrestrial life can't use this stuff. Evidently rice and barley can manage silicon as well. But again, it's not something that is particularly used in our type of life. Here's a picture of diatoms. The electron micrograph there, shows one example of one species of diatom. They have such beautiful little skeletal structures, and I don't know if many of you are familiar with diatomaceous earth. These skeletons of diatoms over time can build up into very large deposits which can be mined. Diatomaceous earth occurs naturally in my laboratory. I have a jar of it, and it helps as an aid to filtration because all the spikiness of the diatom skeletons helps trap tiny particles in whatever I'm trying to filter. So, basically, there's a lot of different ways for, like what I'm talking about here. There's a lot of different ways for organisms to manage silicate. So it seems to have evolved a number of different times, but it's still just not widespread. So here's some pictures of diatoms. So basically little glass skeletons of tiny creatures. And we can actually breed the capability of managing silicon or silicates into terrestrial life. So there's older research here. It was in Science News a while back. But really only selective breeding of 3 generations to put, make silic phthalanes, they're called, so directly pop silicon in the place of a carbon into an organism. Individual replacements of silicon into carbon structures can happen. The resulting compounds tend to be a little bit unstable, and if it gets or these things get less and less stable as you actually attach silicons to other silicon. And there's some reasons for that, which I'll get into. There we go. Yeah, I mean, silicon chemistry is well known. Here's a little molecule. It's basically an artificial amino acid. So this group on the left is your regular template for an amino acid. You have the carboxylate group. I don't know if you can see my little pointer going. Have a carboxylate group. It's got the 2 oxygens and the carbon. That's attached to another carbon where you have also the amino group attached. That's a nitrogen, ostensibly with two hydrogens, but because the nitrogen here is basic and the carboxylic acid is acidic, there's a hydrogen that you would usually think of living on the CO2 group over here. It pops over to the nitrogen. So you end up with minus charge on this end and a plus charge on that end. That structural feature is conserved between all amino acids. What differs between amino acids is what's attached to this middle carbon here. So if they're like something like 20 or so naturally occurring amino acids, and they have things like hydrogen on, or like a phenyl group on, that's C6H6, or C6H5, I should say, or something like SH or S-methyl, methionine, things like that. So, basically, this artificial amino acid. Why would you make this? Well, if you were trying to tag a particular protein with a amino acid or with an amino acid that you could track, for example. So, like silicon is NMR active. It basically can act as a little radio transmitter, and one could um, look at the silicon magnetic resonance signal in a uh, modified protein if one wanted to. So, yeah. Uh, So yeah, basically, looking at what they were doing here, um, I I believe we have a protein here, and I think this is hemoglobin, in fact, and we basically have um, a silicon molecule. Here it's got two within groups near the active site of this particular protein. No, that's not a hemoglobin, but they all look the same to me. This is why I'm an inorganic chemist. Is it in stereo? I think it is. I think I did with cross-eyed stereograms on. So yes, this is a cross-eyed stereogram. So if you happen to be able to see such things, then that pops right out in 3D, and you can see some of the protein in pink in front, and the yellow and the white in back, and then the beautiful colored section kind of shows you electron density, with blue being areas of less electron density, ranging through green, yellow, and then red where there's high electron density. You can see the highest electron density is around the oxygen atom. So, and I can make this talk available so to others so that one can practice with these things. So there we go. So, question is, can you search and replace? No. And that's the end of talk. And thank you very much. And no, that would be evil, because we can talk about why it's you can't just search and replace, right? There's a whole bunch of issues: thermodynamic bonding, the ability of electron transfer. Just aside, let's see. I think someone's got their microphone on. But yeah, so here is DNA molecule, and you just can't cut. You can't just plop in the silicon for carbon. One interesting thing here is, we are silicon or carbon-based life forms, but there's a lot of phosphorus, nitrogen, and oxygen doing some heavy lifting in DNA as well. Um, you know, the bonding to nitrogen, um, for than the bonding between carbon and nitrogen as well. So you have to take into account all of these things. All right. Okay. So bond energy. Um, the units here are kilojoules per mole. What's a mole? Well, it's a big number. So it's 6.02 times 10 to the 23rd. Um, so if you have that many molecules of something, well, you've got a nice chunk you can hold in your hand. If you had 6.02 times 10 to the 23rd, oh, I don't know, stars, well, you'd probably have a universe's worth of stars, or at least an observable universe worth of stars. That's the order of magnitude. Atoms are so small, so they don't um, have um, much mass to them. So you can. It takes a lot to actually make a chunk that you. That's uh, uh, Angel, since your microphone is on, could you shut off? I can hear you washing your dishes. Thank you. Perfect. Okay. So here's the thing. These relative numbers. You can look at a carbon hydrogen. I've got that right up here, 416. Well, a silicon hydrogen is 323. Okay, so quite a bit stronger. A carbon-carbon bond, 346. Silicon bond, 250 to 335, depending on what's attached. Okay, kind of similar orders of magnitude there between carbon and silicon. Right? And you can see like the rest of the numbers here. But one thing here, you look at a single bond for carbon-carbon, 246. A double bond for carbon-carbon. And double bonds are hugely important. Um, the multiple bonding between carbon and nitrogen, and carbon and oxygen, that's one of the things that makes life really work. All of the, let me go back. Backslash one. Nope. Yeah. So looking at these little rings down here in the rungs of the DNA ladder, that's full of multiple bonds. There's at least three double bonds in there. And that happens in every single of the ATCG, all of the bases that make up the rungs of the ladder. These little rings are flat and full of multiple bonding. So if a silicon-silicon bond is worth 222, a silicon-silicon bond double bond is worth 100, right? So notice how a carbon-carbon double bond is about, almost twice as strong as a carbon-carbon single bond, but the same situation for silicon. The silicon-silicon double bond is half as strong as a silicon-silicon single bond. Okay, so that means if you have two silicon-silicon double bonds, right? So 4 silicon atoms in total. You could arrange them in a tetrahedron and form 4 silicon-silicon single bonds, right? So the reactivity of this very weak bond is going to encourage the formation of single bonds and not be something that stays. I do have data here for the carbon-carbon triple bond. Again, it's pretty strong. Silicon-silicon triple bond strength wasn't available. Those are pretty hard to make. I think there are examples of, they would be called silines. But yeah, I don't think anyone's measured their bond strength. The mixed one, silicon carbon, is 163. So again, that's fairly weak. Um, is the bond strength due to the size of the atom? Yes. And let me get right to that. Silicon likes to bond to other elements more than it does itself. Okay, so you're not going to get long chains. That's a good thing to know. Um, and the fancy multiply bonded rings do not persist if silicon is present. So those are the three takeaways. Silicon likes to bond to other things rather than itself. It doesn't like rings. And one consequence down here of the thermodynamics is that notice how strong a carbon-oxygen double bond is versus the silicon-oxygen double bond and single bond. Um, it turns out that because multiple bonding is not favored for silicon, single bonds are more favored. You get silicon dioxide and silicates being kind of a network solid, and CO2 is happy being a small molecule. So, yeah, so here's the orbitals. There's internal structure to heavy element orbitals. Carbon, you have these things called p orbitals. They're um, basically kind of a higher order resonance of um, where you can put electrons. Let's say for carbon, there's two shapes. One is a sphere, right? Put an electron, it can rattle around inside a sphere. Um, Carbon has another shape where electrons can go, and it has this dumbbell type of shape here. And that dumbbell type of shape, there's three of those that are available. They align along the x-axis, the y-axis, and the z-axis. So what I'm just showing you here is z-axis stuff. If you bring two p orbitals, that's what they're called, on carbons close together, you can get the regions really overlapping nicely. Notice how there's phase differences. There's a green lobe and a red lobe. Lobes of the same color can overlap and allow electrons to flow between the two areas without impediment. Lobes of different colors tend to cancel out and make like a no man's zone, so where the electrons can't go. The orbitals are basically telling you what the probability, they're related to the probability of finding an electron. Mathematically is the square of the number, and it is the square of the orbital that gives you the probability. And if you're combining orbitals, you have to use the actual orbital. Bottom line, two different colors, they overlap, they're going to cancel each other out. You'll get a zero in there somewhere, and there'll be a place where the electrons can't go, and you get repulsion. For carbon, you don't get that. No problems. But because of the internal structure on silicon, if you get the two silicon atoms too close together, you get red and green. You get the internal structure of the silicons interfering with the overlap of the red regions. So here you start to see red and green overlap if the silicons get too close. Bottom line, if silicons get too close, the internal structure of the orbitals that are used for bonding causes repulsion. Huh. That's true for all of the elements except for your second row elements, the p orbitals. The p orbitals can't get too close. Right? So, well, we do have silenes. I'm thinking of Dolly Parton. That's Jolene. We could have a lovely time singing a little song about silene. Please don't take my oxygen atom. we can isolate silenes. Here's a beautiful example. This is a silicon-silicon double bond. And to keep that stable, you have to put a lot of organic cabbage around the silane. Um, so they've got two, I call them ferrocinal groups, and then two, oh, those are phenyl rings with a whole bunch of isopropyls. Essentially, the silene is the silicon-silicon double bond is always stable in that molecule because it can't react with a neighboring molecule to make single bond. The molecule is pretty much just isolated. It'll keep some oxygen away. Keeping oxygen away from compounds is not really a problem in most of the work we do because we just like work with them under vacuum or work with it under argon. But a silicon-silicon double bond will react with itself, like two molecules with silicon-silicon double bonds, if the silicons can get close enough together and can arrange themselves into a tetrahedron, those double bonds can just redistribute to form tetrahedron with just single bonds. Right? So yeah, not what we want. Oops. And there we go. So again, I said carbon has a large supporting cast. I like the way I said that. Can we put put other things into biology? Yeah, we've got HOs, Ns. We've got lots of iron and zinc in us. Potassium, sodium, those are really, really important for us. Calcium is hugely important for us, both in terms of us being a structural component. Our bones have a lot of calcium in, but calcium plays some key signaling functions. For example, helper enzyme to nitric oxide synthase, which makes NO, which is a neurotransmitter. Helper proteins called commodulin, and basically the calcium ion is what activates the commodulin to turn the NO production on and off. So phosphorus instead of silicon. Could we put silicons in the backbone of DNA, maybe, instead of the phosphoruses? Well, we have to look at the bioavailability. So I'm going to inflict the knowledge of a Courbet diagram on you. Stay tuned. What is a Courbet diagram? So, Courbet is going to tell you about acidity and it's going to tell you about H plus and electrons, right? So it's going to tell you whether you're in an oxidizing environment or a reducing environment, and it's going to correlate that with the presence of acid or base, right? And we're going to see that we live in a particular environment if we consider the Courbet diagram to be a map. And where interesting silicon structures could happen is in a different place on that map. Okay? There's interesting silicate chemistry, but it's not really in the range that's compatible with our biochemistry. We could get some more interesting chemistry under quote unquote reducing conditions, like things where electrons are more available. I would say that we live under fairly oxidizing conditions because we have to deal with an oxygen atmosphere, and oxygen likes to suck away electrons and do things. So we're going to have concentration of height of H plus or pH on one scale. We're going to have potential, electrical potential on another scale. Courbet is the name of the scientist, yes. So here's the Pourbaix diagram of iron, for example. So here we have pH, okay? And pH is defined as the negative of the logarithm of the concentration of H plus acid in moles per liter. And neutral water would have, for reasons, a concentration of H plus of 10 to the minus 7 moles per liter. So neutral water has a pH of around 7, right? And we tend to live in environments where things can be slightly acidic or slightly basic. Sort of like vinegar that you get from the store and you put on your French fries, is pH is going to be pH is going to be like 2 or something like that. So that's a little too acidic. You can ameliorate that if you add a little bit of base to your vinegar, you get a buffer solution, and that might get you down to something that's more like a 4. Let's see, your blood is slightly basic, but not terribly basic. And living conditions end up having pH range in about 4 to 9 or so. Be a generous estimate. Um, the potential, and here we just have it listed in volts. And volt, if you remember, the units of volts are joules per coulomb. So it's energy per charge, right? So it's basically telling you how hard it is to remove electric charge, like how much energy you have to invest to remove a certain amount of electric charge. So if you think about iron, iron often is iron 3 plus, as you see in rust. Other forms of iron, there's iron 2 plus in magnetite. There's iron 2 plus in other surface rocks if they're protected from the air. That tends to be the range where we find iron or light in our environment. The dotted lines here are telling us about the formation of hydrogen from water, and the upper one is telling us the oxidation of water to make O2. And there's so much water that ends up in our environment setting the limits of our electron transfer. Because, right, if you get too oxidizing, the water is going to turn into O2, right? So that's something that happens and is managed by green plants through photosynthesis. Right, down here, formation of hydrogen is less common, but can happen in geothermal processes. Particularly if we get some more of these more reduced forms of iron, reduced being they have more electrons. So actual iron solid, not something that you would expect to find on the surface of the earth, unless as a naturally occurring thing, right? Is the vertical axis logarithmic? Technically, yes, because energy, volts and energy have a logarithmic relationship. Do they? No, delta G is energy equals minus NFE, so that's a direct relationship. But logarithms pop up all the time when you're thinking of concentration to voltage. That would be the Nernst equation, which is a whole other atrocity that I can inflict on you at some other time. But yeah, so the bottom line for the y-axis, if you're thinking concentrations, that'll be a logarithmic relationship. Yeah, you know what? Chemistry doesn't use very much volts. All of chemistry can probably be done with a 9-volt battery. As long as you could make electrodes that were specific enough to do a particular reaction. Going back to thinking of bond strengths, one way of thinking of chemical reaction is that you have a model, you have to take apart all of the parts, and then you have to put them together in some other way. You could think of taking apart all the parts as being an investment of energy, and you get a return on that investment once you start putting things together. Honestly, if you think of the transformations that actually happen, the difference is really only a few volts. That's why batteries, a single cell battery, doesn't give you a huge amount of, right? If you take the most oxidizing material and the most reducing material, say like you're trying to make a battery with elemental fluorine and sodium. Okay, well, that's a nightmare waiting to happen. It'll just go boom if you don't manage carefully. But that's probably only going to give you like 5 or 6 volts. And that's as far as you can get with chemistry. Unless, you stack up your batteries into a pile and, get the voltage building up that way. So, yeah, I love logarithms. Yes. So let me go on to the next slide, give us a comparison. So if you think of the Pauly diagram for silicates, where our generous estimates of our normal living conditions kind of is off the diagram here, a lot of the interesting stuff you can do with silicates is like over here in the Drano area of the pH diagram, right? Drano is basically um, aluminum turning is mixed with, I think it's sodium hydroxide. So we have highly reducing conditions and um, highly basic conditions. So that it occurs to me that if you want to make friends with um, a Horta from Star Trek, you bring some Drano along, right? It'd be probably like the same as giving an apple to a horse. So yeah, basically the silicate the interesting chemistry of silicon where you have silicates with hydrogens attached directly to the silicon atoms is way down here. Um, so partly why we don't get interesting like interesting chemistry, biochemistry of silicon is that where it does stuff is not relevant to current conditions on the planet. Are there places where such conditions are more natural? Yeah. Let's see. Oh yeah, one thing I should mention is these guys have octahedral structures around silicon. So each silicon has 6 things attached instead of the 4 that I talked about earlier. Um, our biochemistry is all happy with tetrahedra, but um, 6 coordinate things, octahedra, tend to get rejected, right? And so I have a node up here, hexavalent chromium. So that would be chromium. So well, I'll call it chromate, but it's Cr O4 2 minus. Basically a chromium with 4 oxygens arranged in a tetrahedron and a 2 minus. It looks just like phosphate to our biochemistry. And so it can get into a cell and then it can rip electrons away from things that should not have their electrons ripped away and basically start some bad things. That is why hexavalent chromium is so bad. It basically mimics something that the body would want and then wreaks havoc. let's see. Oh, the silicon structure, not silicon's natural occurring structure is the same as diamond. So when you have any computer chip or anything that is just pure silicon, like your solar panels or whatever, it is the diamond structure. Um, so basically, every silicon is attached to every other silicon. Every silicon is at the center of a tetrahedron of other silicons. So the diamond structure of silicon is actually not particularly rare or anything like that. Um, Oh, and silicon carbide has the same structure as well. Um, yeah. And so yeah, so basically, um, if you think about reducing environments and high pH environments, with more tetrahedral looking things, the Horta would be living down here. And maybe underground where there might be deposits of iron or other metals that could be reducing in contact with water. That at high temperatures, that might be a better zone. Yeah, the care and feeding of your Horta. How to train your Horta. So I also like Dr. Seuss. Where's that Doctor Who? Whatever. Um, so most of these Probet diagrams that we have in the literature for our lab environments, but you would expect the whole environment to be reducing, basic, hot, high pressure, um, iron and carbon. So iron, carbon solid, that would be diamond or graphite. Those exist underground. They can reduce silicates. An aqueous environment with set limits, we probably would not get something like silane or huge silicon molecules or anything like that. Um, but we would have certainly some interesting environments. Definitely high pressure and high temperature where um, carbon might not be able to do the interesting things that it does under our conditions. It might just tend to be oxidized to carbon dioxide. And so down there may be some interesting stuff. Uh, and so, like currently, hydrothermal synthesis is used in labs to make interesting structures, which can do catalysis. This research actually, let me do a segue here to the 2025 Nobel Prize. That hydrothermal synthesis kind of formed the precursor to people making metal organic frameworks, because it was a pain in the ass to try to figure out how a substance is going to turn out after you bake it at 1,000 degrees. It's much easier to figure out how to make a molecule or how to make a structure rationally by taking various connecting blocks and allowing them to crystallize, and then maybe doing some post-processing. Okay. Yeah, so there are lots of known minerals. Basically, if you're going to think about silicon-based life, you're going to have to worry about the tendency of silicates to form minerals. And you will have to assume a very limited supply of carbon. Otherwise, carbon-based life is going to be outcompete any silicon um, based life. I'll show you structure up here. I'm going over a little bit. I apologize for that. You pop this down. So silicates can form extended structures. So what I've done here is, where have I put it? Yeah. So what I've done here is taken a set of tetrahedra and link them together in a helix, right? So these, and I've got a left-handed helix and a right-handed helix. One is red, one is blue, right? So silicates can form helices just like DNA can form helices. Um, and these guys could kind of encapsulate other things. The sphere is supposed to represent maybe I don't know, an iron or potassium or calcium or something like that. These, the red and the blue helix are going to be very highly charged and will need a set of ions to, like positively charged metal ions to stabilize them. Right, so a small chunk might be able to encode some information if um, the metal is right. So 100% speculation there. That's why this is a um, advice to science fiction writers. You wouldn't do the one-to-one replacement as you would with as you would, you know, for DNA, but you could mimic something potentially that does DNA. Here's another example. Let me see if I can just get this down and around. Here is a small chunk of asbestos. I know we're all doing as best as we can. There's a small chunk of asbestos. And asbestos is a silicate, and it's actually an aluminosilicate, which means it's got layers of tetrahedra. So the top layer with the beige tetrahedra, you can see, and then layers of octahedra in green, and at the centers of those octahedrons are um, um, aluminum atoms. And these form extended sheets. So like, think of a plane in, you know, continuing, you know, in 2 dimensions. And these sheets stack up. Uh, there is another way for these things to arrange themselves. They can actually roll into a scroll, right? So, yeah, so sorry about exposing you all to asbestos here. Again, we're hopefully Second Life. The red spheres are just oxygen atoms. So, you know, at every vertex is supposed to be a red sphere. And in fact, inside, oh, okay, well, this is the molecule where I got rid of the insides to make it less of a burden on resources for the region. But inside there will be a silicon atom in the beige, and the green would have aluminum in them. And again, the red, I also think, have silicons. So what can you do with something like asbestos? All right, so again, for the science fiction writers, I think, right, so again, here's the structures of the silicate as tetrahedron, the aluminate as octahedra. You can bind them to make something like asbestos. And I have been talking about these as just shapes, so I'm going to move on with those. We can form the short chains and rings. I showed you that one earlier. And so I showed you this thing a little bit earlier as well, so I'm going to move on with that. Oh, and this is jadeite. I've got that modeled somewhere, but again, I want to move forward. There's all sorts of minerals that you can make with silicates, and just having these tetrahedra sheets separated by sheets of ions and basically sheets of water. This is a chlorapathyllite from the American Mineralogist database. I've got that as a model. But honestly, the DNA style encoding is out because these helices that I showed you, they're just going to stack like a handful of pencils and not be able to wind and unwind. Well, that's why I'm more thinking that if we're going to have a Horta, it's going to do stuff more with the asbestos. Those are the asbestos type that I showed you. I guess red is the magnesium in this structure. So that's synthetic asbestos. Why people would make synthetic asbestos? Well, it gets to this making these types of scrolls. But here you can see how the flat layers would line up. Now these things can form nanotubes or scrolls, as they're affectionately called in the literature. So you can see that these sheets kind of roll up into scrolls. And yeah, this stuff is particularly nasty. In fact, there's been some studies that seem to show that it's iron in the iron impurities in naturally occurring asbestos, which promote, it's called Fenton chemistry. It's basically catalytic oxidation of water to form hydroxyl radicals, which then attack your biochemistry, making, polymerizing your cell walls and just attacking things. And right, so just the, and of course, it's catalytic, so it never stops as long as there's oxygen and water around. So yeah, that makes it very, very nasty. Okay? So these things can roll up, and we can make all sorts of different types of them synthetically, all sorts of these little scrolls. And exactly how they roll up does depend on mechanical and chemical factors. It's really, if you take some of the magnesium and change them into different elements, you're going to inject some stress into the flat sheet that tends to cause it up, or cause it to roll up. Right? So you can think of injecting different elements to cause different stresses in a sheet, right? And, figure out how to encode information by injecting those different elements. Again, this is for the science fiction element. So, maybe we put an iron atom in somewhere instead of an aluminum. Well, iron can do iron 2 plus and iron 3 plus. There's a redox control there that might change the size of an iron atom, that might change the stress, causing maybe in one oxidation state a tendency to be flat, or in another oxidation state a tendency to roll up. Right? Maybe we can control the flat versus rolling up with some chemistry. Right? Carbon life right now has had 2 billion years of evolution to figure all of its stuff out. So if we're going to kind of give some advice to the science fiction folks, we need to keep it simple. So far, what would that look like? We still need to move stuff around. We still need to encode information. Maybe we can do that in with scrolls instead of chains like DNA is. But can we unroll a scroll? Well, my suggestion would be that we'd have vanadium around, because vanadium 3 plus likes to be octahedral. Vanadium 4 plus is often, we call it square pyramidal. So it's like that last corner pops off, and that might loosen things up enough to allow some scrolling to happen, or the unrolling to happen, right? Now we can do a picture somewhere of how I would envision this. Water transition metals, well, they undergo electron transfer fairly easily. They're fairly readily available. Way back when, I showed you a picture or a table with some availabilities. Vanadium was at 0.01 terrestrially. Carbon was 0.03, so that's comparable. Um, so I'm also not sure if Copper Plus and Copper 2 Plus could do the same thing, but Copper is pretty much available as well. All right. And then here's a little picture. One could think of a scroll unwinding and then silicates and maybe some helper molecules popping in to do some replication, depending on which way scrolls could unwind and replicate. And then if some chemistry had to happen, maybe there could be areas where a scroll kind of partially unwinds and opens up and does some stuff. Right? Harkens back to the interesting sorts of chemistry that our zeolites and artificial zeolites were doing right at the beginning of my talk. So they're nice scribbles. Basically, how do we assemble little tetrahedra things into a sheet, into a complementary sheet? I'm then ripping off what DNA does in one dimension and trying to envision what that does in two dimensions. So that's sort of how I would think how a Horta could be built. So thank you, Todd. I drew that by hand while I was on a plane coming back from a conference, just thinking about how this would go. So yeah, it's took me a long time. Um, so I think that's where I wanted to end up in for part one of the talk, right? So if silicon is going to outcompete carbon, carbon doesn't have to be around, right? So we have to have an exceedingly low carbon, like almost a no carbon environment. So this is what I'm thinking underground, high pressure, high temperature. The carbon's going to be in the form of diamonds or carbon dioxide. If water's around, diamonds are nice because they will be reducing agents and allow the silicon to do interesting and maybe under the right conditions, aluminosilicates and has some chemistry that might initiate self-catalysis, right? The replication that would be necessary for something to be a self-replicating system that can evolve by Darwinian evolution. And hopefully we would, if we were looking for this stuff, we might look for things that have things that are like asbestos-like scroll type things with defects that are apparently random but are apparently like identical, something that we couldn't explain. So yeah, so moving on, I think that's where I am. Where's Bert? Poor Bert. He passed away a few years ago. I lived until he was about 19 years old, so he had a good long life. So thank you, everyone, members of the Science Circle, all of my colleagues and staff at SIUE, the National Science Foundation has been very supportive of Science Circle. It's a wonderful venue for doing science outreach. So I'm hoping to continue to be able to do that, and obviously my plaques for the patient. So I think that is the last slide other than weird stuff. Yeah, we're not going to talk about that. That's part two. We're going to talk about ammonia as a solvent in January when I do part two of this talk again. So apologies that I couldn't keep up with the chat so much. When I talk, I kind of have to think about my inner world a little more. But I'd be happy to answer any questions if people have questions. Also love to wish everyone who celebrates a happy Thanksgiving next week.